Bismillahirrahmanirrahim. Today we are gonna study the covering of your brain called meninges and the part of your brain, the cerebral cortex. In the picture, you are looking into the brain and the spinal cord, and you should know that these both structures are protected inside the skull and the vertebral column. And why they are soft and gelatinous? Because they have got less intercellular connective tissue, right? Now we have to discuss the meningeal covering first. You see from the top that there lies the scalp with the hairs on it. Then you can see the periosteum next to it. Then comes the skull bone. Now we have to see the three meningeal covering of your brain, right? The outermost is the dura mater with its two layers. We will discuss later on. Then you can see there is a dark purple color layer, that is the arachnoid matter, marked by the green arrow. And then you can see the lighter or mauve color layer called the pia mater, marked with the blue color, right? Now we have to discuss the outermost meninge called the dura mater, right? You can see that it has got two layers. The external called the periosteal layer, just adherent to your bone of the skull along with its periosteum, right? Then it has the inner meningeal layer. The color being the darker blue is the periosteal layer and the light blue is the meningeal layer. And if we talk about that, there is a epidural space, which is a potential space present in between the periosteal layer of the dura mater and the bone of the skull. This epidural space is actually present inside the vertebrae around your spinal cord, and there it contains a plexus of thin walled veins and the areolar connective tissue, right? And what else you can see here, that the two layers of dura mater that encloses venous sinuses. Here you can visualize the superior sagittal sinus, right? And the dura mater word means the tough mother. So we expect that it consists of dense fibroelastic connective tissue, right? Next to that dura mater, you have got another layer in purple color is the arachnoid mater, which is avascular layer because it doesn't have any nutrient capillaries for it, but it allows passage of the blood vessels to pass through it. And we can see next to dura it is present, and we can say dura mater is always separated by the arachnoid mater through a subdural space, right? And we expect the internal surface of all the dura mater is having the simple squamous epithelium, which is next to the dura mater is another thin layer called the arachnoid mater. This meninge is present just below the dura mater, and we expect dura mater is always separated from the arachnoid by a thin subdural space, right? And the internal surface of all dura mater, and as well as when it is present in the spinal cord, is being covered by the simple squamous epithelium of mesenchymal origin, right? And the subdural space normally does not have any content, right? But after any injury or any other pathological condition, fluid can be collected, right? Now concentrate on the arachnoid mater. We will move towards other picture. Here in this picture that you can visualize the skull, the dura mater and the lighter shade arachnoid mater, which we should focus. This is a thin membrane, no doubt, but is spider or web-like structure, right? You can see it has got two components. The upper component is sheet-like, isn't it? And the lower component is showing the trabeculae or septa made up of collagen, and the cell's fibroblast could be a part of it. And it is continuous with the third inner meninge called the pia mater. And we have told that this arachnoid mater is avascular, but as you can see that it is giving a passage for the vessels to run in, right? and in between these trabeculae or septa you can see the large sponge-like cavity called subarachnoid space, and it is filled with the CSF, right? And what is the purpose of this fluid filled space? That basically provides cushions and protects your CNS, right, from any minor trauma. And the subarachnoid space, which we have mentioned, is filled with CSF. It has to communicate with the ventricles of your brain, right, where CSF should be produced. That should be kept in mind. And we have mentioned that it lacks nutritive various capillaries, but give passage to the larger blood vessels you can see in the picture. And we also expect that if it is the arachnoid matter around the spinal cord, it should be showing fewer trabeculae, right? And there we can more clearly distinguish it from the pia matter. And another important fact about it, this arachnoid matter and pia matter, they are often associated, and they are forming often a single membrane, and there we call them as pia arachnoid or leptomeninges, right? Here's something more about your arachnoid matter. You can see that in few areas that arachnoid is projecting or protruding into the two layers of the dura mater, and this is a place where venous sinuses are being formed, and these are blood filled, you would say, venous sinuses, and the CSF filled protrusion of the arachnoid layer that are being now covered by the vascular endothelial cells lining the sinuses. These are called as arachnoid villi, right? What is the function of these? They have to absorb the CSF, and they have to absorb this CSF into the blood of the venous sinuses. So they are the site for absorption of CSF, right? It must be clear now. Now below that turquoise color layer, the pia mater, which is a tender mother. A name is taken from its meaning. So it's a very delicate layer, and it consists of flattened, mesenchymally originated, flattened, modified fibroblast, right? And simple squamous like epithelial cells it consists of. And it, you can see, covers whole of the entire surface of your CNS tissue, right? The pia does not directly contact nerve cells or fibers, right? That is very important. So we should know that it is separated from the nervous element below by which thing? By a very thin superficial layer of astrocytic processes, and we call them as glia limitans, right? And that should be adherent firmly to the pia. Means these two elements are firmly adherent, then the pia matter and that layer of astrocytic end, that is forming now the physical barrier through the astrocytic feed and the pia matter. So separating your CNS from the CSF in the subarachnoid space, right? Here in this picture, we are supposed to see another function, you would say, of the pia mater. You are looking into a slide above and the schematic representation below of the choroid plexus. Okay, what is choroid plexus? Basically, it is a highly specialized tissue that shows folding in many villi, and you can see them projecting into the ventricles. So there are 4 ventricles of the brain, right? So they are present in the roof of the 3rd and 4th ventricles, and along with that the 2 lateral ventricles, right? And the ependymal lining here is directly in contact with that of the pia mater, right? Now you can see that each villus of this you would say choroid plexus, it contains a well vascularized pia mater covered by, you can see in the picture below, by the cuboidal ependymal cells, and there is the pia mater next to these cuboidal cells, and it is made up of, you can see, the flattened squamous shaped cells, right? And the function of choroid plexus is to form CSF by removing water from the blood and releasing it as CSF, right? This CSF, which is present where? It fills all of your ventricles and the central canal of spinal cord, and you find it in the subarachnoid space, perivascular spaces, right? And if we talk about the content of the CSF, it contains sodium, potassium, chloride ions, right? But a very little protein could be there, and the only cells that could be present in a very less amount could be the lymphocytes, right? And we should know that the CSF, it is providing ions, and why? They should be required for CNS neuronal activity, and in the arachnoid they have to help absorb what? Mechanical shocks, right? And if the CSF is produced by the choroid plexus, what is the structure that is going to absorb them? The arachnoid villi, which we discussed already, they are the pathway for the absorption of your CSF back into the blood through the venous circulation. Another important thing in relation to that of your CNS tissue, that it doesn't have any lymphatic vessels, right?